wabarakatuh. My name is Nur Arif Aiman bin Muhammad Saleh.
My name is Muhammad Afham bin Mustafa.
My name is Muhammad Akil Benyamin bin Ahmad.
We are from class S1ET4.
So today we are going to teach you a video about transcription in protein synthesis subtopic.
Enjoy this video.
Get it?
Hello everyone. Our lesson for today is transcription process, which is the shortest topic in this chapter. We are really sure that this topic is really easy to understand. So, without wasting any time, let's proceed to the learning. Enjoy this video. So, to kick off, let's begin our adventure with the definition of transcription. Do you know the real definition of transcription? Transcription is a process where genetic information in DNA is transcribed into an mRNA molecule. Transcription occurs in cytoplasm for prokaryotic cell, while for eukaryotic cell it occurs in nucleus. Why does the location is different? It is because prokaryotic cell does not have nucleus and the DNA is located in cytoplasm. The transcription process in prokaryotic cell is simpler than the process in eukaryotic cell. In the end of this video, we will explain the reason why there are differences between these two cells. In the first place, we should know how many stages are there in the transcription. Do you know how many stages are there? There are 3 stages that occur in the transcription process, which is initiation, the first stage, followed by elongation, and closed by termination stage. Initially, in easy word to understand it better, initiation is the starting process. Firstly, the RNA polymerase, which is the enzyme that played a main role in the transcription process, it will bind to initiation site of DNA that is called promoter site. When the RNA polymerase attached to the promoter site, it will unwind and unzips the DNA, and one of DNA strand is used as a template for transcription. After initiation, the elongation of mRNA strand occurs. The RNA polymerase will be used again to add nucleotide to the 3 end of the growing strand. The direction of the growth of the RNA molecule is 5' to 3' as it continues along the double helix. I want to include an important note, so please give me full attention, class. When transcription takes place, as RNA is synthesized in 5' to 3' direction, DNA template is read in the opposite direction, which is 3' to 5'. Then the process is continued with termination stage. After elongation, the growth of the RNA molecule continue to proceed. Only when RNA polymerase reach the termination site on the DNA, the process will stop. The sequence of bases that marks the termination site will signal RNA polymerase to stop adding nucleotides. So what is those sequence of bases? There are three triplet bases of stop signal, which is UAA, UAG, and UGA. So after the RNA nucleotide was stopped to be added, the mRNA strand and RNA polymerase are released. The mRNA transcript is released and leaves the nucleus into cytoplasm. The released RNA transcript is now called primary RNA transcript, or in other words, it is called pre-mRNA. However, this only associated with eukaryotic cell, as there is no pre-mRNA in prokaryotic cell. As we said earlier in the video, the transcription process in prokaryotic cell is simpler compared to eukaryotic cell. In prokaryotic cell, there are no nucleus. So, mRNA produced by transcription is immediately translated without additional processing. Meanwhile, in eukaryotic cell, there are presence of nucleus. The nucleus provides a separate compartment for transcription, the original RNA transcript, which is called pre-mRNA. Pre-mRNA is processed in many ways before leaving the nucleus as mRNA. Original transcribed pre-mRNA from eukaryotic cell consists intron and exons, while prokaryotic cell consists only exons. So, what is introns and what is exons? Introns is non-coding sequences, while exons is coding sequences. This relates to the explanation earlier that mRNA produced by transcription is immediately translated, as the mRNA is functional because it has a continuous coding sequences, which is exons. As eukaryotic cell consists intron and exons, in mRNA processing, it changed the RNA after transcription. An mRNA that is ready to be translated will be formed as the changes happen. Hence, how does this happen? An enzyme called spliceosome excises or cut the introns and ligate or join the exons in a process called RNA splicing. This will form functional mRNA, which contain a continuous coding sequence. The mature mRNA is transported to the cytoplasm through nucleus pore. It is ready to be translated in translation. That's all from us. Thank you.